Today we are going to talk about genome mapping. All of us know a map is a place where we have landmarks. For example, you want to travel from a place from Delhi to Mumbai, you'll be having a lot of milestones which act as landmarks to let you know where you have reached. Similarly, when you're talking about a map on the genome, we are talking about certain landmarks that we define a particular genome with. So that is what is genome mapping. Genome mapping defines landmarks on the genome or certain features on the genome which are quite specific for the genome. Right. Now, why do we actually need a genome map? Initially, when people were doing sequencing the genome means they used to ideally sequence the entire genome. The human genome map, which started in 1990, completed in 2013, used only Sanger sequencing. And the technique that was used is mainly shotgun sequencing. And this shotgun sequencing, if you see the figure down below, which I'm currently explaining, shotgun sequencing. Here, the whole genome is broken down into pieces of 500 base pairs, and each of the 500 base pairs is sequenced, and the assembly is actually done based on the overlaps that you get across these 500 base pairs. The major drawback of this entire sequencing is when you are using short fragments of sequences, completing the genome gets really difficult. Even if you have certain contigs that you have completed, per se, that you have completed certain contigs like these, that they are seen in the figure here, right? To place the contigs exactly onto a particular place onto the genome, you need landmarks that are already there on the genome. For example, here, you can clearly see on this particular genome, there are landmarks from A to G, A, B, C, D, E, F, and G. Now, on each of the contigs that you have identified, we first identify whether that contig contains any of these landmarks. For example, here, in this particular contig 1, we have a landmark E, let us say. And in this particular contig that you have here, we have a particular landmark F and G. Now what happens? In this particular contig, you have the landmarks B and C. Now, having got these landmarks, now what happens? It becomes very easy for us to exactly place these contigs onto the exact position in the genome. For example, this E fragment, the contig 1, the so-called contig 1, now goes into a place here. And the contig 2 here goes into a place here on the genome. And similarly here onto the genome. And likewise, having got the defined landmarks on the genome, we can place these segments, short segments that have been sequenced in order so that we get a continuous assembly. But still, getting a continuous assembly is really difficult with short segments. So the first basic purpose why we need a genome map is to assemble the genome in order, in particular order. And if we don't have these landmarks, we can't assemble the genome because we do not know whether a contig 1 comes first, contig 2 comes first, or contig 3 comes first. Here, exactly, the contig 3 has come first because of the positions of B and C, and then the contig 1, and then the contig 2. Now, when I say a contig, you should understand that it is the assembly of two overlap sequences, 2 or more overlap sequences. Now, having got the first purpose why we actually need a genome map, the first is to assemble the genome, and the second one is to take care of the repetitive sequences. Take care of the repetitive sequences. Now, when I talk taking care of the repetitive sequences, we'll say that there are two copies in the genome, right? Here there is one copy, and here there is the other copy. Now, these copies, when sequenced as short fragments, you'll have fragments like these, and you have an overlap between this fragment and this fragment. Similarly, you'll have an overlap between this fragment and this fragment. But, however, these two sequences being similar, there is every chance that this overlap here is not overlapping with this in the course of your assembly, but may go and overlap with this sequence because of the similarity in the sequence. So likewise, here what I had shown here is, like, I had shown the overlap between these two sequences. The sequence here, the sequence here, and the sequence here, which is completely wrong because these are not from the continuous set of fragments that we have sequenced. Actually, it should be an overlap with the fragment here, but it has overlapped here, and you get an incorrect overlap. Now, having got a genome map, knowing the flanking sequences of this particular fragment and this particular fragment and these fragments here around, you know the flanking sequences, then this problem of having an incorrect overlap can be solved. Having known these, for example, you know that in this particular overlap, there is D here. Now what happens? Now when you know that there is D here, a part of the D here that is sequence, you know that this particular segment has to come here and map to the fragment that is adjacent to D here. And similarly, now you have a part of the fragment that is E here. Now, in the process of assembling, these two fragments now come in a correct overlap, whereas this problem of incorrect overlap can be corrected just because we have the landmarks D and E. Having said all this, right? Having said all this, right? The current NGS technologies and the algorithms that are run, right, will take care of all these problems. And people are now assembling the genomes, right? I don't say without the need of the maps, but with the minimum need of the maps, right? But still, I'll tell you why with the minimum need of the maps, because when we come to NGS, we'll discuss that in detail, but people are still using a technique called as optical mapping in assembling the genomes. We'll discuss that later on. But however, here, the most important thing that we'll have to discuss here is, considering that NGS is in vogue nowadays, and people are not using the maps that like the way they have used earlier, right? We cannot say that the maps are not needed at all. Listen to me. Even in NGS, you need a map for the purpose called as functional annotation. For the purpose called as functional annotation. Having known a set of landmarks on a particular fragment, sequencing a particular fragment or identifying a particular fragment, right? You can make out what this fragment exactly contains and then functionally annotate depending on the genes on the fragment or the genes present adjacent to the fragment. You can functionally annotate this and assign a function to this particular fragment and then correlate with the phenotype of interest. So what exactly are we talking about? Here we are talking about having got NGS with all the algorithms in place. Don't ever think that the map is obsolete or mapping is obsolete. Mapping has still got a major role in functional annotation. In this functional annotation, when it becomes a part of in the course of my lectures, I'll be also telling you what functional annotation is in NGS and how this mapping would help us in actually functionally annotating a particular fragment identified, right, and sequence, and how this fragment identified can correlate with our phenotype of interest, we'll have to we'll have a look at. Right. Now, now we'll broadly classify gene mapping into two major categories. Gene mapping is basically classified into two major categories. And what are those categories? The first one is genetic mapping, and the other one is physical mapping. First one is genetic mapping. The first one is genetic mapping. And the next one is physical map. Physical mapping. Now, genetic mapping, all of us know that this is basically dependent upon Mendelian genetics and commonly done using plant experiments. And also, this can be done by knowing certain pedigree details in humans. So this can as well be done in animals. If we know the pedigree details, this as a separate section we'll be dealing later on, and we'll be majorly talking about linkage mapping. So today, we'll first discuss about physical mapping. Under physical mapping, right, you have certain broad categories which we'll discuss. Physical mapping can further be classified into, the first one is restriction mapping. First one is restriction mapping. And the next one is optical mapping. Optical mapping. And then you have certain physical techniques like the first one is fluorescent in situ hybridization. And then you have certain techniques like STS mapping. So these are the major categories of mapping if you look at. Further, optical mapping can be classified into gel stretching, molecular combing, and the one that is currently in use nowadays is bio-nano. We can further classify fish into further subcategories. So fish can now be classified into, we're talking about fish can be further classified into one, metaphase fish and fiber fish. Metaphase fish and fiber fish. We'll talk about each one of them in detail in the next talk.